So, hi, I'm going to be a junior this year, and I studied immunotherapy. So, immunotherapy is currently emerging as the fourth most prevalent cancer treatment. The aim of our study was to assess how different kinds of cancer therapies can be combined with boosting cross-presentation. So the main topic of my poster, dendritic cells. Dendritic cells are the most effective antigen-presenting cell. So what is cross-presentation? So first, the dying tumor cell releases signaling molecules to attract dendritic cells. Number one, this is the tumor cell that's dying, and these are the dendritic cells that have received the signal and have gone to the site of the tumor. D is combination of dendritic cell therapy with immunogenic cell death inducing drugs. The most effective combinations were cross-presentation enhancement methods, namely the dendritic cell vaccines applied along with radiotherapy.
Great job that you have learned so much and done this work using a review paper. So you had given a conclusion that radiotherapy seems to be the best for combination with immunotherapy. Why do you think that is?
One of the studies that I read found that radiotherapy, the success is very dependent on the number of dendritic cells inside the tumor. So let's say that a tumor is treated with radiotherapy, but there are very few resident dendritic cells. The radiotherapy will not be as effective because the dendritic cells are what initiate the immune response after it's treated. So if we either enhance the number of dendritic cells inside the tumor or the functions of the dendritic cells that are already there, the immune response after radiotherapy can be much stronger.
Yeah, that's right. That's a very good understanding.
I'm a first year undergrad student, and I'll just be talking to you about this project, which entails discussing the impact of experimental, clinical, and biological variables on EGFR expression in head and neck squamous cell carcinoma. So what is HNSCC? Well, it's known as head and neck squamous cell carcinoma, and it's the 7th most common type of cancer worldwide. But just a little bit more about the cancer. It's a group of cancers affecting the mouth, sinuses, nose, or throat, and that's illustrated in this image. As far as the methodology and experimental design goes, we first conducted some preliminary research utilizing NCBI and some published journals. And eventually we were able to obtain some background information on HNSCC and EGFR, for example, risk factors, incidence, mutations, etc. So these are the results of our t-test. The two graphs represent the results for the specific sample size. So the graph on the left shows the relationship between the mean EGFR expression and the mean age. Based on this bar graph, a higher mean expression of EGFR occurred in the population above the mean age in this given sample. So based on this graph, the R value and P value, you can deduce that relatively strong positive linear correlation between the linear copy number and the EGFR expression were fairly strong. So just to wrap it up in the conclusion, I wanted to reiterate the results. So based on our analysis, which had some limitations, some of the results provide support to existing theories in certain published journals. So for example, the t-test yielded significant results for the following variables in the sample size for the EGFR expression and patients above the average age and below the age, as well as EGFR expression and patients with and without a history of alcohol consumption over a period of time. Thank you for listening. I'm open to any questions you may have.
Did you find some other cancer type where this correlation exists and where the mechanisms for this correlation, like causation, is described?
Yes. So with this data, you're not really able to imply causation because given that HNSCC is unpredictable, there's several variables that could influence the results, which could skew the p-values.
Project effect of TP53 mRNA expression on glioblastoma. So we'll start with an introduction. So glioma is a common type of brain tumor, and the term is used to signify the tumor's origin in the glial cells. So the gene of interest for this research is TP53, which is a tumor suppressor gene which codes for the protein P53, also known as the guardian of the genome. When a mutation in the TP53 gene happens, it affects multiple signaling pathways. The objectives of the research was first to sort through the publicly available data to check for TP53 mRNA expression. Next was to study TP53 mRNA expression in relation to the fraction of genome altered in patients. And lastly was to compile the data and see how TP53 affects glioblastoma patients as a whole. So the data set that I used was the Firehouse legacy from TCGA. And so now we'll go into the tests. So this is the role of TP53 in patient data, TP53 relative linear copy number, and TP53 mRNA expression. There is a strong inverse correlation. They'll come to my future direction and discussion, as well as the conclusion. In a cancer cell, the cell cycle is disrupted through a mutation in the p53 pathway. And since the younger patients have a higher ability to repair cell damages, they have higher TP53 mRNA expression. And so in a possible future variation of the project, I would have to design the study with more exclusion criteria, and the sample size will need to be more specific.
Very nice presentation. I must appreciate you, like the way you have added the background. It shows like you're really into this brain tumors and brain research. So yeah, I loved it.
Hi, and as Dr. Manuel said, I will be a junior in high school, and my research project was on the promoter methylation of HTRT and CDKN2A in head and neck squamous cell carcinoma, which can be abbreviated as HNSCC. Epigenetics is basically when gene expression is controlled, but the gene itself is not altered, where mutations mean that the gene itself is altered. But epigenetics is when the expression is altered externally. So methylation is an example of that, and that's when methyl groups are added to the gene, and they cause less expression usually for that gene. So the two genes that I looked at were HTRT, which stands for human telomerase reverse transcriptase, and CDKN2A, which is a tumor suppressor gene that is heavily involved in the cell cycle checkpoint system. And my hypothesis was that HNSCC patients have promoter hypermethylation of both CDKN2A and HTRT. And so the two independent factors that had the most effect on HNSCC were age and HPV status. This scatter plot shown here shows that there is a weak negative correlation between HTRT methylation and mutation count. However, it is significant because the p-value is below that critical value of 0.05. Unlike mutations which have already affected the genome, methylation can be changed with therapy, and so it's an important field to study for studying cancer treatments. And then for future studies, we might include analyzing a different cohort and running similar tests to increase that sample size and get more significant results. Or we could look at the methylation of a few other genes, such as DNA repair genes, and see how those affect HNSCC to see whether methylation could be used as a biomarker for this disease.
Finding the correlations with methylation of promoter region. So because expression of the gene depends pretty much on methylation of promoter region, the others can be methylated differently, but this one is important for the functioning. So this is where you can find more reliable results and more of a correlation. That's just a suggestion. And that is a great job that you've done. Thank you.